what we had talked about last week was that when you're going into the cell cycle, going out of G0 into G1, there's going to be mitogens like platelet-derived growth factor that are going to bind to their receptor, tyrosine kinase receptors. That's going to do the ERK pathway and through AKT pathway, PI3K, AKT pathway is going to induce transcription of cyclin D. And then once you get cyclin D made, cyclin D is going to bind to its kinase partner, CDK4 or CDK6. This is going to form an active kinase complex, and cyclin D, CDK4 or 6 is going to phosphorylate an inhibitory protein called retinoblastoma protein that's binding to the transcription factor E2F. So it's going to partially phosphorylate RB, slightly relieving the inhibition of E2F so that E2F can start inducing transcription of cyclin E. And then cyclin E, which is the next cell cycle protein that's important, is going to be transcribed. It'll be made, it binds to its partner, CDK2. So you have now an active kinase complex, and then cyclin E is going to fully phosphorylate RB on other residues that are on RB. That's going to fully relieve E2F inhibition, and now E2F can transcribe the genes like cyclin A, for example, that you need to get the cell out of G1 and into S phase, so this G1S transition. Okay. So here's another technique that I want to introduce to you of how we test in the lab or learn through the lab how the cell cycle is working. This is called immunoprecipitation, and what immunoprecipitation allows us to see is inside of the cell what proteins are directly interacting, or even indirectly, but interacting with each other. So the way it works is, let's say here, this is a complex protein of interest, complex of proteins. So say that's cyclin D and its CDK4 interactor. This, you would grow cells on a plate, you would isolate the protein from the cells. This is a test Eppendorf tube here. So these are proteins that you've isolated from the cell. And what you're interested in is the cyclin D interacting, what it's interacting with in the cell. So you'll take an antibody, for example, the cyclin D, add it to that protein lysate, and then pull out, using that antibody, you pull out cyclin D, and anything that cyclin D is still bound to is going to be pulled out. That's the immunoprecipitation part of this assay. And then you separate off everything that hasn't bound to the cyclin D, for example, complex, and then you take this complex and you can run it on a Western blot gel, a Western blot to see what cyclin D is bound to. Okay. All right, so that is what was done here. So this is done for, in this experiment, what they did is they took cells and they transfected cells with different plasmids. So they're looking, in this case, they want to demonstrate that indeed RB is bound to E2F. Okay, so that's what they're, and they're also demonstrating that the release, so remember, the way that RB relieves binding is no longer binding to E2F, is once RB gets phosphorylated either by cyclin D, CDK4/6, or cyclin E, CDK2. So here in the cells, they have E2F inside of the cells. They have what's called a dominant negative CDK2, so this is a CDK2 that doesn't work. So even if that CDK2 binds to cyclin E, it's not going to form an active complex. Because it's not going to form an active complex, it can't phosphorylate RB, okay? So that's the important part here. So they do an immunoprecipitation. They're going to IP for E2F, so they're going to pull out E2F and anything that E2F is bound to. In the case of this dominant negative cyclin D, you see that you end up immunoprecipitating or pulling a lot of RB out of that complex. And that's because that RB is still stuck to E2F, okay? This dominant negative CDK2 was not able to phosphorylate RB, and so it stays stuck to E2F. And now in the absence of that dominant negative CDK2, now you see less RB is actually bound to E2F. Does that make sense? Does anybody have questions about how that works? Okay. All right, now we talked about the luciferase assay last time. Just to remind you the way this works, is you take a promoter of interest. So let's say you're interested in looking at the cyclin E promoter, seeing if that cyclin E gene is turned on. You take the promoter for cyclin E, you fuse it to a gene called luciferase. You put that plasmid into cells. When the transcription factor, in this case we care about E2F, when E2F binds to that cyclin promoter on this plasmid, it's going to drive expression of luciferase. So the cell is going to make the luciferase enzyme. You isolate the protein from that cell that's going to include this luciferase, and you add the substrate for luciferase into this test tube. And based on the amount of luciferase that the cell made, that's how much fluorescent signal you're going to get. So the more transcription you have going on here, the brighter the signal is, and you can read this on a plate reader. Okay. So in this experiment here, going back to the luciferase assay, so what they did is that same dominant negative CDK2. So they are showing here, and in this case they're looking at E2F transcriptional activity. So what they did here, I was showing you before, like you could use a promoter for cyclin E, for example. We actually know the sequences that these transcription factors bind to. So you can take like the E2F promoter sequence binding sequence and fuse that to luciferase. And so that's what they did here. So they have a E2F promoter, the sequence that transcription factor would bind to. They fuse that to luciferase. And what they see is under normal circumstances, when you don't have this CDK2 that is inactive, you get a nice level of luciferase activity. So that's telling you that this E2F transcription factor is active. However, if you put that dominant negative CDK2 into the cells, remember this is the guy that's not going to be functional, which means that RB will not get phosphorylated, which means that RB stays stuck to E2F, so E2F can't transcribe genes. And so that's why you have this low level of luciferase activity. Okay? Okay, so based on that, what would you guys predict would happen here? So this is going to be, in this experiment here, this is a E2F immunoprecipitation, so they pulled E2F out of the cells. What they did in this case, they have a mutant RB, so this is an RB that cannot be phosphorylated, okay? So that tells us since it can't be phosphorylated, it's going to be stuck to E2F. So when you do the immunoprecipitation in this case, what do you expect? Do you think the band is going to be darker or lighter than that first one? Yeah, it's going to end up being darker because more of the E2F, if this RB can't be phosphorylated, it stays stuck to E2F. So you're going to end up immunoprecipitating that out. And then if you put in a rescue construct, so if you put in now a normal RB, you'll see that the signal goes back down. You pull down less RB. So here, when you have a mutant RB that can't be phosphorylated, so it stays bound to E2F, when you immunoprecipitate E2F out of the cells, you're going to get more RB being pulled down, and then you can rescue it with now a wild type of normal RB, and now that guy can get phosphorylated, and here you see you pull down less E2F. You pull down less RB. Okay, same paper. They did the luciferase assay, so what do you expect is going to happen with luciferase? This is looking at E2F transcription activity. What's going to happen in the case of the mutant RB? You're going to have higher or lower luciferase activity? Yeah, it's going to be lower because, again, it's going to be lower because this guy can't get phosphorylated, so it stays stuck to E2F, so E2F can't function. And then again, you can rescue this phenotype by adding in a wild type RB, and so now you get luciferase activity.